Hello students, today we'll learn about vaccines. First, a brief introduction. Vaccination protects a recipient from pathogenic agents by establishing an immunological resistance to infection. An injected or oral vaccine induces a host to generate antibodies against the disease-causing organism. Therefore, during future exposures, the infectious agent is inactivated, that is neutralized or killed. Its proliferation is prevented and a disease state is not established. Now let us see the mechanisms of vaccine preparation. Several factors must be kept in mind in developing a successful vaccine. Some of the factors are: the development of an immune response does not necessarily mean that a state of protective immunity has been achieved. What is often critical is which branch of the immune system is activated, and therefore vaccine designers must recognize the important differences between activation of the humoral and the cell-mediated branches. The second factor is the development of immunologic memory. For example, a vaccine that induces a protective primary response may fail to induce the formation of memory cells, leaving the host unprotected after the primary response to the vaccine subsides. Now coming to the type of vaccines. The first one is the whole organism vaccines. The first one is attenuated vaccines. Microorganisms can be attenuated so that they lose their ability to cause significant disease, pathogenicity, but retain their capacity for transient growth within an inoculated host. Attenuation often can be achieved by growing a pathogenic bacterium or virus for prolonged periods under abnormal culture conditions. The advantages of attenuated vaccines are: because of their capacity for transient growth, such vaccines provide prolonged immune system exposure to the individual epitopes on the attenuated organisms, resulting in increased immunogenicity and production of memory cells. As a consequence, these vaccines often require only a single immunization, eliminating the need for repeated boosters. The second point, the ability of many attenuated vaccines to replicate within host cells makes them particularly suitable for inducing a cell-mediated response. One example is the Savin polio vaccine. It consists of 3 attenuated strains of poliovirus and administered orally to children on a sugar cube or in sugar liquid. The attenuated viruses colonize the intestine and induce protective immunity to all 3 strains of virulent poliovirus. Savin vaccine in the intestine induces production of secretory and EA, which serves as an important defense against naturally acquired poliovirus. The vaccine also induces IgM and IgG classes of antibody. Unlike most other attenuated vaccines which require a single immunizing dose, the Savin polio vaccine requires boosters because of the 3 strains of attenuated poliovirus in the vaccine interfere with each other's replication in the intestine. With the first immunization, one strain will predominate in its growth, inducing immunity to that strain. With the second immunization, the immunity generated by the previous immunization will limit the growth of the previously predominant strain in the vaccine, enabling one of the 2 remaining strains to predominate and induce immunity. Finally, with the third immunization, immunity to all 3 strains is achieved. The disadvantages of attenuated vaccines are: the possibility that they will revert to a virulent form. The rate of reversion of Savin polio vaccine (OPV) leading to subsequent paralytic disease is about 1 case in 2.4 million doses of vaccine. This reversion implies that pathogenic forms of the virus are being passed by a few immunized individuals and can find their way into the water supply, especially in areas where sanitation standards are not rigorous or where wastewater must be recycled. The second point, a serious complication with the first Salk vaccines arose when formaldehyde failed to kill all the virus in 2 vaccine loads. which cause paralytic polio in a high percentage of recipients. The third point is, attenuated vaccines also may be associated with complications similar to those seen in the natural diseases. The second type of whole organism vaccine is the inactivated vaccines. Another common approach in vaccine production is inactivation of the pathogen by heat or by chemical means so that it is no longer capable of replication in the host. It is critically important to maintain the structure of the epitopes on surface antigens during inactivation. Heat inactivation is generally unsatisfactory because it causes extensive denaturation of proteins. Thus, any epitopes that depend on higher orders of protein structure are likely to be altered significantly. Chemical inactivation with formaldehyde or various alkylating agents has been successful. The Salk polio vaccine is produced by formaldehyde inactivation. The advantages of inactivated vaccines are: the first point, attenuated vaccines generally require only one dose to induce long-lasting immunity. Killed vaccines, on the other hand, often require repeated boosters to maintain the immune status of the host. In addition, killed vaccines induce a predominantly humoral antibody response. They are less effective than attenuated vaccines in inducing cell-mediated immunity and in eliciting a secretory IgA response. The second point is, even though they contain killed pathogens, inactivated whole organism vaccines are still associated with certain risks. A serious complication with the first Salk vaccines arose when formaldehyde failed to kill all the virus in 2 vaccine loads, which caused paralytic polio in high percentage of recipients. Purified macromolecules. Some of the risks associated with attenuated or killed whole organism vaccines can be avoided with vaccines that consist of specific purified macromolecules derived from pathogens. 3 general forms of such vaccines are in current use: capsular polysaccharides, inactivated exotoxins, and recombinant microbial antigens. Coming to the first one, bacterial polysaccharide capsules. The virulence of some pathogenic bacteria depends primarily on the antiphagocytic properties of their hydrophilic polysaccharide capsule. Coating of the capsule with antibodies and/or complement greatly increases the ability of macrophages and neutrophils to phagocytose such pathogens. These findings provide the rationale for vaccines consisting of purified capsular polysaccharides. Example, Streptococcus pneumoniae, which causes pneumococcal pneumonia, consists of 23 antigenically different capsular polysaccharides. The vaccine induces formation of opsonizing antibodies and is now on the list of vaccines recommended for all infants. The vaccine for Neisseria meningitis, a common cause of bacterial meningitis, also consists of purified capsular polysaccharides. The disadvantages are their inability to activate Th cells. They activate B cells in a thymus-independent type 2 (TI2) manner, resulting in IgM production but little class switching, no affinity maturation, and little, if any, development of memory cells. One way to involve Th cells directly in response to a polysaccharide antigen is to conjugate the antigen to some sort of protein carrier. For example, the vaccine for Haemophilus influenzae type B (HIB), the major cause of bacterial meningitis in children less than 5 years of age, consists of type B capsular polysaccharide covalently linked to a protein carrier, tetanus toxoid. The polysaccharide protein conjugate is considerably more immunogenic than the polysaccharide alone, and because it activates Th cells, it enables class switching from IgM to IgG. The next one is toxoids. Some bacterial pathogens, including those that cause diphtheria and tetanus, produce exotoxins. These exotoxins produce many of the disease symptoms that result from infection. Diphtheria and tetanus vaccines, for example, can be made by purifying the bacterial exotoxin and then inactivating the toxin with formaldehyde to form a toxoid. Vaccination with the toxoid induces anti-toxoid antibodies, which are also capable of binding to the toxin and neutralizing its effects. Condition for production of toxoid vaccines must be closely controlled to achieve detoxification without excessive modification of the epitope structure. The problem of obtaining sufficient quantities of the purified toxins to prepare the vaccines has been overcome by cloning the exotoxin genes and then expressing them in easily grown host cells. In this way, large quantities of exotoxin can be produced, purified, and subsequently inactivated. The third type is recombinant techniques. A number of genes encoding surface antigens from viral, bacterial, and protozoan pathogens have been successfully cloned in bacterial, yeast insect, or mammalian expression systems, and the expressed antigens used for vaccine development. Example: Hepatitis B vaccine. Hepatitis B vaccine was developed by cloning the gene for the major surface antigen hepatitis B virus HBsAg and expressing it in yeast cells. Recombinant vector vaccines. Genes that encode major antigens of specially virulent pathogens can be introduced into attenuated viruses or bacteria. The attenuated organism serves as a vector, replicating within the host and expressing the gene product of the pathogen. A number of organisms have been used for vector vaccines, including vaccinia virus, the canary pox virus, attenuated poliovirus, adenoviruses, attenuated strains of salmonella, the BCC strain of Mycobacterium bovis, and certain strains of streptococcus that normally exist in the oral cavity. One example is the vaccinia virus. The attenuated vaccine used to eradicate smallpox has been widely employed as a vector vaccine. This large complex virus with a genome of about 200 genes can be engineered to carry several dozen foreign genes without impairing its capacity to infect host cells and replicate. The genetically engineered vaccinia expresses high levels of the inserted gene product, which can then serve as a potent immunogen in an inoculated host. Like the smallpox vaccine, genetically engineered vaccinia vector vaccines can be administered simply by scratching the skin, causing a localized infection in host cells. If the foreign gene product expressed by the vaccinia is a viral envelope protein, it is inserted into the membrane of the infected host cells, inducing development of cell-mediated immunity as well as antibody-mediated immunity. The DNA vaccines. Plasmid DNA encoding antigenic proteins is injected directly into the muscle of the recipient. Muscle cells take up the DNA and the encoded protein antigen is expressed, leading to both a humoral antibody response and a cell-mediated response. The DNA appears either to integrate into the chromosomal DNA or to be maintained for long periods in an episomal form. The viral antigen is expressed not only by the muscle cells but also by dendritic cells in the area that take up the plasmid DNA and express the viral antigen. The fact that muscle cells express low levels of class I MSC molecules and do not express co-stimulatory molecules suggests that local dendritic cells may be crucial to the development of the antigenic responses to DNA vaccines. The advantages of DNA vaccines are: the encoded protein is expressed in the host in its natural form. There is no denaturation or modification. The immune response is therefore directed to the antigen exactly as it is expressed by the pathogen. It induces both humoral and cell-mediated immunity. To stimulate both arms of the immune response with non-DNA vaccines normally requires immunization with a live attenuated preparation, which introduces additional elements of risk. Cause: prolonged expression of the antigen, which generates significant immunological memory. Refrigeration is not required for the handling and storage of the plasmid DNA, a feature that greatly lowers the cost and complexity of delivery. One disadvantage is the only protein antigens can be encoded. Multivalent subunit vaccines. These vaccines employ solid matrix antibody antigen SMAA complexes by attaching monoclonal antibodies to particular solid matrices and then saturating the antibody with the desired antigen. By attaching different monoclonal antibodies to the solid matrix, it is possible to bind a mixture of peptides or proteins composing immunodominant epitopes for both T cells and B cells to the solid matrix. These multivalent complexes induce vigorous humoral and cell-mediated responses. Their particulate nature contributes to their increased immunogenicity by facilitating phagocytosis by phagocytic cells. Another means of producing a multivalent vaccine is to use detergent to incorporate protein antigens into protein micelles, lipid vesicles called liposomes, or immunostimulating complexes. Mixing proteins in detergent and then removing the detergent from micelles. The individual proteins orient themselves with their hydrophilic residues towards the aqueous environment and the hydrophobic residues at the center so as to exclude their interaction with the aqueous environment. Liposomes containing protein antigens are prepared by mixing the proteins with a suspension of phospholipids under conditions that form vesicles bounded by a bilayer. The proteins are incorporated into the bilayer with the hydrophilic residues exposed. Immunostimulating complexes, ISCOMs, are lipid carriers prepared by mixing protein with a detergent and a glycoside called QuIL-A. Example: membrane proteins from various pathogens including influenza virus, measles virus, hepatitis B virus, and HIV have been incorporated into micelles, liposomes, and ISCOMs and are currently being assessed as potential vaccines. Now coming to the conclusion. We have learned that a state of immunity can be induced by passive or active immunization. Short-term passive immunization is induced by transfer of preformed antibodies. Infection or inoculation achieves long-term active immunization. Three types of vaccines are currently used in humans: attenuated, avirulent microorganisms, inactivated, killed microorganisms, or purified macromolecules. Protein components of pathogens expressed in cell culture may be effective vaccines. Recombinant vectors, including viruses or bacteria engineered to carry genes from infectious microorganisms, maximize cell-mediated immunity to the encoded antigens. Plasmid DNA encoding a protein antigen from a pathogen can serve as an effective vaccine inducing both humoral and cell-mediated immunity. Realizing the optimum benefit of vaccines will require cheaper manufacture and improved delivery methods for existing vaccines.